So if this is your first time in our class, listen, you have to put in your score that you want on the board, on a piece of paper, okay? So make sure you write that score that you really want to score on the exam, and that's your target right there. If you don't write your score down, if you don't have this piece of paper lying inside your house somewhere on your, everywhere where you study, you put it on the wall where you brush your teeth next to your mirror, listen, that's your target. It gives you a sense of purpose so you know exactly what you're shooting at. Even if you miss it, you're gonna get close to that score. Remember, you're gonna get tons of bonuses for this lecture, right? You're gonna get lecture notes. You're going to get the insider secrets to scoring 260, my exam ritual book. Remember, you have my direct line. My email address is dradesina@ftplectures.com. My phone number is 856-258-0572. You can call me if you have any questions. I'm here to help you as long as you're part of our course. And you can email, go to our website, smashusmle.com, or email us at admin@smashusmle.com. And that's our phone number, 215-465-3339. All right, that's our direct number for the company. Just call and Paula will be able to help you. All right, let's start high yield step 1 hematology oncology lecture, okay? So before we begin, we need to understand the big picture, all right? This is what I always teach you guys. There's a lot of minutiae information for the boards, but always look at the entire forest while not nagging at the leaves next to the trees, okay? If you see the big picture, you'll be able to understand. What are we learning here today? We are learning about the blood. That's it. If you know that the bone marrow produce, blood cells, all right, whether it be, B cells, or whether it be also be like, your white blood cells and your red blood cells and your platelets, all that good stuff all come from the bone marrow, right? The bone, every bone in your body pretty much produces this stuff. And also understand the spleen is involved in any hematologic condition. We'll talk about how the spleen is very important in clearing red blood cells and stuff like that, okay? And the thymus gland. Those are the 3 organs basically that's doing this in your body as we speak right now, okay? Hey, thank you for watching this video. Listen, I'm gonna be giving away a free ebook on the insider secrets on how to score 260 on the USMLE. Make sure you watch this video to the end because I'm gonna be giving you a link to be able to download it. And the be it all end all is when everything goes well in physiology, right? You should have normal red blood cells. You should have normal white blood cells. You should have normal platelets, okay? Because they all have different functions. They're like this host of army, right? That are responsible for keeping us safe. They're our immune system. They're also our, UPS truck that carry oxygen around our bodies to keep us alive. That's pretty much the bottom line of what they do, okay? And when they die, they have to go somewhere and be cleared out. Okay, and that's the bottom line. When something goes haywire, we develop hematologic conditions, right? We develop cancer of the blood, all right? And when you get cancer of the blood, that's bad. We'll talk about different cancers of the blood in this lecture. The other side of it is that we have blood flowing through all our capillaries and our veins and our arteries. And if you cut your skin, you're gonna cut a blood vessel. When that blood vessel bleeds, right, it's gonna ooze out. Well, the body's got to keep you alive. So the body's like, we're not going to let you die. So we're going to prevent you from dying. So the blood will clot. It's a good thing, right? Because the blood will, if you hold pressure on the wound stop bleeding. And all of a sudden you're like, wow, I stopped bleeding. That's great. Guess what? If anything happens to that pathway that allows your blood to basically clot together, you're screwed. Or what about this? There's always a balance, right? Your blood has to be thin, and also has ability to clot. So it has regulatory mechanisms that makes it basically not clot. So it tells you, okay, don't clot our blood because this blood needs to be flowing. At the same time, it knows when to clot when there's a bleeding, and it knows when to not clot. So that's really important to keep you alive, or else everybody will be having strokes and MIs and mesenteric ischemia, and life will be bad, okay? So that's basically the bottom line of this lecture, right? So you need to understand it's a bleeding problem or cancer of the blood, and then everything else is supposed to go well, all right? So let's look at a hematopoiesis, right? Hematopoiesis, how do we generate all these hematopoietic cells, right? Well, every cell comes from their grandmother, okay? Their grandmother is this dude called the multipotent stem cell, right? Stem cells basically have the ability to do whatever the heck they want. They're pre-programmed, and basically you can have 2 ways to go as a stem cell, right? You can go the myeloid stem cell pathway, all right? So if you go you can go the myeloid stem cell pathway, or you can go the lymphoid, okay, pathway, Okay, so here you see, let's go down to lymphoid real quick. Okay, in the lymphoid, we want to make basically lymphoblasts, all right? It's called lymphopoiesis, right? Now, this lymphoblast has 3 ways to go. We can either make a B cell, okay, or we can make a T cell. That's a cell. Oh, like, dude, you're gonna become a T cell. It's like, awesome, thank you. And this other one can become a natural killer cells. That's it. Those are the 3 kind of blast, cells that initially start. But the thing is, these B cells can become transformed into plasma cells. Now, plasma cells are important because they make antibodies, right? IgM, IgG, IgA, right? IgD. So they're really, really important for you to be able to, when you activate them eventually, these plasma cells will basically fight infections because you make all these antibodies that you guys always talk about. Like, you've memorized so many antibodies, and when you guys learn about, different side of hypersensitivity reactions. Oh, IgG binds to complement and neutrophil activation, and we get a type 3 hypersensitivity. Well, it's coming from the plasma cells, all right? So then we have T cells, right? I love me some T cells because T cells, they're helper T cells. And then we have the cytotoxic T cells, all right? And we're not talking about immunology here, so we're not gonna really go into details of exactly how these guys really work with, being a CD4 or CD8 cells, but we also have these natural killer cells, right? The kill cells, all right? And that's it. Like, if a cell is not acting right, like, just kill it so it doesn't turn into cancer, all right? That's it. That's literally it. That's one lymphoid stem cell. That's what it can do. Now, on the other side is the myeloid stem cell, right? So if you look at the myeloid stem cells, this is where we get to make red blood cells, right? So look here. The first thing is erythropoiesis. How do we make a new red blood cells? How is the new red blood cell going to be born? Well, a new red blood cell is going to start as an erythroblast, all right? And that erythroblast is in the bone marrow, right? All of these guys in the bone marrow. But in the bone marrow, we make a small little reticulocytes. That's a little baby, a little teeny baby red blood cells, who eventually is going to make a mature red blood cell that's going to be shoot out from their bone straight up into your blood. It's like, dude, you are officially ready to go. You're going to be in a red blood cells, you're an erythrocyte. Let's go carry some oxygen and take CO2 back out of our tissues so that we can continue to live. So, right? So that's great. However, we need platelets, right? So for platelets, we're going to need megakaryoblast, right? Megakaryoblast, right? So megakaryoblast forms a megakaryocyte. Cyte means a cell, right? So which eventually just makes your platelets, right? Platelets are very, very important. Listen, we're going to learn a lot about these platelets today, all right? These platelets are going to go into your bloodstream, right? When they get to the blood, they're responsible for clotting, primary hemostasis. And then we have the granulocytes, right? See this granulocytopoiesis, right? So you're going to need a myeloblast, right? And this myeloblast has the ability to differentiate into an eosinophils, right? Eosinophils, basically, these guys are responsible when you have an allergic reaction or you have helminth infections like tapeworm infections. These guys are usually activated. If you have bad asthma, your eosinophil count will be very, very high, right? And then we have this basophils, right? Basophils, are going to be broken down, be differentiated from myeloblasts. And then we have these bands, right? So these bands are immature neutrophils, right? So basically, for example, that band is an immature neutrophil in the bone marrow, but when it's differentiated and it's already ready to go into the blood and mature, it becomes a full neutrophil. And we know neutrophils are very, very important, right? They got this multi-lobal cells right inside of them, and they have myeloperoxidase and all this good stuff to kill bacteria. Like, neutrophils are the soldiers. They're coming after you, buddy. Okay. Now, as you can see here, we're going to talk a lot about neutrophils later. All right. And then we have the monocytopoiesis, right? Monocytopoiesis, which is going to initially start as a monoblast. And that monoblast is going to differentiate when you get into your blood as a monocyte. But the monocyte inside the blood has to penetrate into your tissue and become a macrophage. So remember when somebody had a myocardial infarction, right? We always talk about these macrophages that come around after the tissue has died to eat up all the bad tissue, right? Let's clean it up, right? Let's clean this place up. And then we have scar tissue after having an MI, right? These are the macrophages. Also, like all the bacteria that come in, they're going to get eaten up by macrophages, okay? So that's it, guys. That's it, all right? So this is your hematopoietic tissues, all right? And we need to know all this, and we're going to learn about their function, how they work, and all the things we need to know about each one of them, okay? All right. So let's do some Q&A about blood and cellular components, all right? So what are the anucleus cells that are found in the peripheral blood, right? Anucleus cells. Anucleuses, they have no nucleus. Well, it's your red blood cells, guys. Red blood cells do not have a nucleus. Wow, really? Yes, they don't have a nucleus because they don't need it, okay? So remember, guys, I need you guys to be involved in this lecture. The more you participate, the more you're gonna learn, because I'm gonna be testing you guys a lot today, and I need you guys to respond, all right? That's how you're gonna maximize your learning education in this lecture. So also, thrombocytes, which are basically, basically platelets, are small cytoplasmic fragments that don't have a nucleus. So those two of all those hematopoietic cells that I told you guys earlier, they don't have a nucleus, right? So they don't have DNA to make RNA, to make protein, right? Whatever comes out as a reticulocyte that becomes a mature red blood cell, that's it. You're screwed up. That's why they don't last long, right? Because there's no replication inside the cells of these cells. There's no, okay. Well, guess what? If the cell is damaged, it's time for you to move on, okay? Same thing with platelets. Now, what is the lifespan of the different cellular components of blood, right? How long does a red blood cell last before it dies? Like, it works so hard and it's exhausted. What do you guys think? Anybody? Great job, great job. 120 days, that's right. That is the lifespan of a red blood cells, right? It works so hard, carries oxygen from the lung, and then goes to the tissues like, hey guys, take your oxygen, give me some CO2 back, and then take CO2 back, and that's all it does all day long, all right? Okay, so, and then we have thrombocytes, right? Platelets, look, your platelets only last 8 to 10 days, and then we gotta make new ones from megakaryocytes. So you have a higher turnover of platelets than red blood cells. However, white blood cells usually have a long lifespan, all right? Especially in the peripheral tissues, they lasted much, much longer, okay? Now, how is the energy produced in red blood cells? How do red blood cells make energy, right? I told you they have no nucleus, right? Okay, so if they have no nucleus, they don't, do they have a mitochondria? No, the red blood cell has no mitochondria. So how does red blood cell have the energy to be able to make ATP and move along, right? We need that ATP to kind of maintain that sodium potassium ATP is pumped, right? Or else we're gonna swell up and explode, right? So what happens is in red blood cells, they undergo glycolysis, right? That's it, glycolysis. The reason is because they don't use oxygen, right? Imagine, think about it. You have a red blood cell it's an oxygen carrier, right? That oxygen binds to the iron on that's sitting in that protoporphyrin ring that's hanging around the alpha and the beta globin proteins. But guess what? It can't even use the oxygen that it's carrying, which is amazing. It's like somebody gave me food and I cannot even take a bite out of it, but because they're incapable of aerobic metabolism. They have no mitochondria. These guys are gonna basically go through glycolysis, and that glycolysis is gonna be converted into pyruvate, right? And make lactate, all right? Which makes sense because they have no mitochondria, they have no nucleus. These guys are just doing their little thing there, all right? And then they just basically carry oxygen to your tissues. So they only need a little bit of amount of ATP from glycolysis, and that ATP allows them to power them to be able to keep moving on, all right? So what is the main function of erythrocyte? We talked about it. They just carry oxygens from your lungs to your peripheral tissues and CO2 from peripheral tissues back to the lungs for elimination. Like you guys think about it, you guys been taking a nice deep breath in and out of this lecture, and your red blood cells are already doing this for you every second. All right, so from what do thrombocytes come from? We talked about them. They come from the cytoplasmic fragments of megakaryocytes. I always repeat a lot in this lecture, not because I just love to, because repetition is the mother of all learning, guys. So the more we repeat, the more you learn, okay? So which body organ has up to 1/3 of our platelet pools, right? One of our organs in our body is carrying 1/3 of our platelet. Does anybody know? Like literally, 1/3 of your platelet are sitting somewhere inside your body right now. Anybody think about it? I see the answer. Good job, Nana. Mojo Faitimi, awesome. This spleen, right? This spleen is where almost 1/3 of your platelets are sitting inside, okay? All right, and we said that the job, primary hemostasis. Primary hemostasis. Now, if you don't know what primary hemostasis is, we're gonna talk a lot about that in this lecture, okay? So don't worry. But listen, what happens is when you activate a platelet, it's usually when there's an endothelial injury, okay? Platelets are gonna aggregate with other platelets. They're gonna, hey dude, you come over here, man. Let's kind of hang out together. We gotta keep this guy from bleeding out, right? And they interact with fibrinogen, and they will form a platelet plug. Okay. Once they form the platelet plug, they actually contain a dense granules, right? They have adenosine diphosphate, calcium. They have alpha granules, okay? So these granules of platelets, the way you remember is that they all have F in them, all right? This granule has von Willebrand factor, has an F at the end of it, fibrinogen, start with an F, fibronectin, and they have platelet factor 4, right? The F in the factor. So that's how you remember all those granules that sit inside of your platelets, all right? These are all important, guys. These are really, really important, okay? So, you know, we will talk about primary hemostasis in a minute. So what are leukocytes? We talk about leukocytes. These are basically your neutrophils, but 60% of your white blood cells are neutrophils, okay? 30% of them are lymphocytes, all right? And then 6% are monocytes, right? Remember the monocytes become macrophages, all right? And then eosinophils makes about 3%, and basophils is only about 1%. So remember, neutrophils play an important role in acute inflammatory response. They help fight bacterial infections. Macrophages basically eat up bacteria. I love it. They just take a bite. They're like, wow, that's yummy, right? You know, poor little bacteria walking around to get a bite in the butt, start crying. And they cause, you know, they do cellular debris. I told you about that when you have a myocardial infarction. Neutrophils are the first guy that's going to come first, right? Because acute inflammation, you have ischemia to your myocytes, right? So remember in that first 24 hours and then 24 to 48 hours, you start getting all these neutrophils come in. Well, they cause that acute inflammation, and then macrophages is going to come in later. They're going to take all that cellular debris and then also eat old red blood cells. Man, these guys are always hungry. They got a big tummy, okay? Now, eosinophils defend against helminth infection. We talked about that. But basophil and mast cells are mediate allergic reaction. Why? Because remember, in immunology, we talked about type 1 hypersensitivity, okay? And in type 1 hypersensitivity, what happens? You have IgEs sitting on top of your mast cells, right? Those IgEs, once they pick up any antigen, they're gonna meet up and kiss each other, and they're gonna cause degranulation by mast cells to release what? What gets released by mast cells in type 1 hypersensitivity, guys? Anyone? Good job. Histamine release. That massive histamine release is going to cause edema. It's going to cause pruritus, itching in the patient, right? And that's why, you know, you see these mast cells are involved in allergic reactions. Same thing as basophils, okay? So that's why you need to understand this. Like, you don't need to memorize it, because if you understand immunology, you'll be able to know, like, oh, mast cells, basophils, these are allergic reactions. They are involved in allergic reactions. See, that's why I always go back to explain the physiology and pathophysiology of diseases to you, because then you'll memorize less of how things function, okay? All right, so what are B lymphocytes and what is their main function? Well, B lymphocytes, they come from the stem cell. That's why they come in the bone marrow. They mature there, but they produce plasma cells and memory B cells, okay? They are important in humoral immunity. Now, what are plasma cells? I told you plasma cells that come from B lymphocytes after they've been exposed to an antigen, right? So you get a virus, a bacteria inside your body, okay? They recognize this. You make these B lymphocytes, which are basically not gonna, you know, make plasma cells, because now they have to remember that the bacteria has been there in the past, and they make antibodies. So next time you get exposed, you can already have another immunity against these organisms, okay? So what are T lymphocytes? What are their function? Well, they mediate cellular immune responses. They originate from the bone marrow, and then they migrate to the thymus. They're like, dude, pack your bags, get out of the bone marrow, we're going to the thymus, all right? That's where we're gonna go get matured. We're gonna go to puberty when we get into the thymus, right? So although they're coming from the bone marrow, and then as you see the name T, right? So they're called T lymphocyte, right? Because the T in their name. So 80% of the circulating lymphocytes are T cells, all right? These are your lymphocytes. Remember those CD4, CD8 cells when we talk about HIV, right? All right, those guys get matured inside your thymus gland, all right? So if you have thymic aplasia, you got a problem with T cells. You can't fight infections, right? So that's why I try to tell you guys, you know, sometimes you guys are memorized. I'm like, listen, if I have thymic aplasia and my thymus is missing, guess what? I'm screwed, because none of my T cell will ever make it to the thymus, because they get to the promised land. They're like, hey, dude, the promised land was never existed. And all of a sudden they're just floating around immature, they can't function. And then when you get infections, guess what? You are screwed, all right? All right, so let's look at some red blood cell shapes and clinical significance. And you guys better tell me exactly what you see right here. All right, now I just put up a slide. I don't know if you guys can see very well. What do you think that is? It's got little spikes around it. It's got that yellow arrow on that little peripheral slide. Does anybody know what that is? So I see Burr cell, Burr cell, Burr cell. So those are acanthocytes. Acanthocytes, so those are spur cells. They have spikes on them, so just little spikes. That's usually seen in liver disease, all right? So when you have liver disease, you see spur cells. Very, very important that you guys know that, because the way hematology works is this, right? So there are particular shapes. Remember, a red blood cell is a small little biconcave cell that kind of floats around and can bend through tissues. Any different shape other than that, there's a pathology associated with it. So when you guys are studying for your boards, they will give you these shapes, and these shapes is really to give you an idea that something's wrong with that red blood cell. Something bad is going on in the red blood cells, and you need to recognize them, okay? What about this one? Here we got another slide with the arrow pointing to it. It's got little dots, like kind of little blue dots. It's almost like a little faded donut with little blue dots. Anybody know what that is? So that's basophilic stippling. Basophilic stippling. That word should be ringing in your head. Every time you hear that, you think about lead poisoning, lead poisoning. You have to know that. That is a very high yield factor of the USMLE. They love me some basophilic stippling, all right? This is a patient that's gonna have lead poisoning. We're gonna talk about a full case of lead poisoning later in this lecture, but I kind of just wanna kind of whet your appetite with some of the shapes because they have clinical significance, right? So if a pathologist is looking at a slide and he sees a bunch of basophilic stippling, it's like, hey, this is a kid that has lead toxicity. This kid would develop mental retardation, developmental abnormalities, abdominal pain. This patient will have kind of lead lines on their bone, all right, on their growth plates, right? This is a serious pathology, right? So you need to know this for your USMLE. They love to test it, okay? How about this one? Okay, what do you guys think this is? Yes, we have a little slide here. It's like somebody's crying. Why are they crying? You know, it's amazing. Like I make stories out of this. I'm like, wow, look at this. So what do you guys think the pathology is associated here? I see Dr. Nguyen here, and we have, what do you guys think? If you see a red blood cell that's crying, it got squished by a fibrous tissue, right? That is a teardrop cells, okay? That's myelofibrosis, guys. Good job, good job. You know, I see Dr. Wayne got that right. Perfect, perfect, good job. So basically when you have a, fibrotic bone marrow, remember, all this reticulocytes got to come from the from the bone marrow. As they try to squeeze through all this net, that's scar tissue, they get basically their body shredded, and by the time they come out, basically half of their body has been kind of zipped out. And then they come out like a little teardrop. Like it's literally the red blood cell has been crying, going through all this like net, it's like a web of a spider web that's so hard. And it's literally crying to get out. And it comes out like, oh my God, I barely made it through. Poor little red blood cell. It's like, yeah, dude, you do come through a bad scar tissue. That's a dry tap, right? So remember that. That's very high yield. And also thalassemia, you can see teardrop cells, okay? All right, how about this one? I love this one. All right, poor little guy, eating a donut, he got bitten in the buttocks. Little red blood cell got bitten in the buttocks, and it's like, oh my goodness, who bit me? What did I do? I was just minding my own business. What do you guys think this is? All right, what pathology is associated with this red blood cell? He got bit. It's like, dude, stop biting me, man. What are you doing? All right, I see a couple of answers there. Good job, guys. Bite cells, bite cells. Those are bite cells, all right? So that is seen in glucose 6-phosphate dehydrogenase deficiency, okay? We will talk about all this pathology. This is just to whet your appetite, get you to some clinical pathology because they are super high yield. Listen, if there's a pathology associated with something, we're gonna test you on the board exam. I guarantee you that. How about this one? How about this one? Anyone want to take a guess at this? Got those little red blood cells. They don't look good. They don't look good. What do you guys think? Right there with the black arrow. I'll put my mouse on it so you can see it right here. Just little red blood cells here, right here. So those are spherocytes. Those are spherocytes, okay? That's what you see in hereditary spherocytosis, okay? And you see autoimmune hemolytic anemia and drug hemolytic anemia. We're gonna talk about that in a minute. You're gonna learn a lot about spherocytes in this lecture. And how about this one? This little red blood cell right here. It's got a little round looking, doesn't look right. Doesn't look right. So those are schistocytes, all right? Schistocytes are seen in patients with TTP, all right, HUS, and HELP syndrome, schistocytes, okay? And then last but not the least, how about this one? What do you guys think? Oh, you're right. I'm actually looking at that. I think I switched that. This slide, this is supposed to be the schistocytes, and this is supposed, this is the spherocytes. This is this, okay. Guys, this slide right here, let me just highlight that. That got switched. I made a mistake there. This is actually a spherocyte, okay? This is a spherocyte, and these are schistocytes. See how they kind of shared? Okay, so let's just make sure, okay? Sorry about that. So, this slide is switched. All right. So with spherocytes, you see hereditary spherocytosis, like I said, autoimmune hemolytic anemia, drug-induced hemolytic anemia. And with schistocytes, right, they shared, that's in TTP, HUS, and because they're mechanically damaged. We'll talk about that in HELP syndrome. What about this last slide here? What do you guys think? This slide right here. That's sideroblastic anemia, guys. Excess iron in the mitochondria, okay? Those are ring sideroblasts. They have all those is a Prussian blue stain, and you see all that mitochondria be stained, okay? It's a lot of iron in there, okay? That's perinuclear ring. And you find out in vitamin B6 deficiency and iron H toxicity. So if you take iron, remember when we talked in micro, we learned about iron H, isoniazid. And if you give a patient isoniazid, they're going to develop peripheral neuropathy because they do not, if you don't give them vitamin B6, all right? This is what you're going to see in iron H toxicity or vitamin C B6 deficiency, all right? That's basically from sideroblastic anemia, all right? So good stuff, good stuff. All right. Now let us talk about some bleeding disorders, okay? Now, guys, this is very, very important. How do we approach a bleeding patient? So they will give you a case of bleeding patients on the board exam, and you need to have a system, all right, of how you approach all your patients that have bleeding disorders on the USMLE. Very, very important, okay? If you don't have a systematic way of approaching bleeding patients, you're always gonna, and that's where I try to explain to you guys that, try to have a system. Like if you have a system, it really makes your life really, really easy, okay? So here's where we're going to approach bleeding patients. There's two ways we're going to approach bleeding patients. We are going to understand that it could be a platelet disorder. Okay. So Harry's asked, how do you differentiate with basophilic stippling? Okay. Let me show you guys real quick. So Harry's asking a question. Look at basophilic stippling. Inside of the red blood cells, there are tiny little donuts, right? They look like little tiny blue dots inside the donuts. So if you ever buy a Dunkin' donut, see, it's like little crumbs on the inside right there. Let me kind of annotate here. See that? There are little dots inside the donut. But if you look at sideroblastic anemia, the entire cell, you see that? You see that? And then you see that central pallor, like the inside of it is kind of pale. Here, the entire thing is basically stained with Prussian blue. That's too much iron, okay? Can you see that, Harry? That is how you differentiate between sideroblastic anemia, all right, from a patient that actually just has basophilic stippling, all right? That's the difference. Faded central pallor with tiny little basophilic stippling, little dotted. That's what you see there in lead poisoning. But vitamin B6, you can see the difference now. There's no central pallor in sideroblastic anemia. All right. So we're talking about how we're going to approach a bleeding patient. Anytime you have a patient that's bleeding, you always have from now and forevermore, I need you to split your brain into two. It's either a platelet problem or a coagulation problem. let me repeat that. Whenever they give you a case of a bleeding patient, always think, is this a platelet problem or is this a coagulation problem? Okay? So let's go through this. This is really, really fun. All right. Anytime you hear the word petechiae, okay? Anytime you hear the word ecchymosis, anytime you hear the word palpable purpura or superficial skin bleeding, right? That is a platelet problem, guys. Petechiae, purpura, ecchymosis, petechiae, purpura, ecchymosis, petechiae, purpura, ecchymosis. Those are superficial or skin bleeding. Guess what? They are non-blanchable. Remember I showed you guys a video. I don't know if you guys saw my video. We had a case of a patient that had roseola the other day in my emergency department that I showed you guys. And I showed you, when you put your thumb on a rash, if it turns white, that's a blanchable rash. If it doesn't blanch, that's a platelet problem. Very, very, very important clinically significant finding on any patient that you see a rash on. Put your thumb on the rash and see if the rash disappears. If the rash disappears, it's a blanchable rash. If it doesn't disappear, guess what? It's either a petechiae, all right? If it's still tiny little dots, or if it's a purpura or ecchymosis, all right? That is a big deal, okay? Now, if they're telling you on the boards about your patient has a mucosal bleeding, mucosal bleeding, well, mucosal bleeding is gonna be a menorrhagia, right? A woman with a heavy menstrual period, or nosebleeds, epistaxis. Whenever you hear the word petechiae, purpura, ecchymosis, menorrhagia, or epistaxis, think platelets. Platelets. This is a platelet problem. It's got to be a platelet problem. There's nothing else that will give me that pathology. That is how I want you to split your brain from now and forever and ever. Don't ever forget this, okay? Platelet problem, all right? And what are you going to first order? You're going to order a CBC, a complete a CBC count, right? And once you order your CBC, the first thing you want to look at is the platelet count. Now, there's a couple of things that's going to happen, right? What you're going to find out is, well, if a patient has petechiae or they have purpura or ecchymosis or they have a nosebleed, well, when you, on a complete blood count, if their platelet count is low, they have thrombocytopenia. Well, now, let's talk about bleeding time. So we don't do this as much anymore, but back in the days, what we used to do is that we'll cut you with a blade, and you have an intern that's got a little clock, and it press start, and it counts how many seconds or how many minutes your bleeding takes until it stops, all right? And if your bleeding time is really prolonged, we say, oh, he has a prolonged bleeding time, right? So what happens is, if I don't have platelets, right? If my platelet count is low, right? My bleeding time will be high. Remember, platelets are involved in primary hemostasis. Now, there are four pathologies that you need to know that will cause me to have low platelet counts. Like I can have ITP, idiopathic thrombocytopenic purpura, okay? Or I can have TTP, or I can have HUS, hemolytic uremic syndrome, okay? So, or I can have HIT syndrome, which is heparin-induced thrombocytopenia. Those are the four things that will cause my platelet count to be low, or it's gonna always make my bleeding count high, okay? So this is how I always think about it. I'm like, oh man, low platelets, okay, is this patient have ITP, TTP, or HUS? Or if the patient is given heparin and 2 days later, we check their platelet count, it's low because you make heparin antibodies, they eat up all your platelets, and guess what? You're screwed. Your platelet count drops from giving you heparin. So you develop heparin-induced thrombocytopenia. Hey, thank you for watching this video. I hope you truly enjoyed. Listen, if you want to watch more videos like this and get a free copy of my Insider Secrets for Scoring 260 on the USMLE, this book is packed with all the secrets that the MBME doesn't want you to know. I want you to click on the link below to be able to download and get a copy. Also, don't forget to subscribe to my channel. You're awesome. Thank you for watching.